Let me, I'll open up. Promptly at noon. So, judging from the large audience, I mean, everyone knows who they're coming here to see. It's my pleasure to reintroduce John Calarco, who many of you probably remember from his time here as a Bauer Fellow up to a couple of years ago. So John is a real Canadian. He was an undergraduate and graduate student at the University of Toronto. We got interested in C. elegans neurogenetics, molecular genetics. Got his PhD working with Ben Blanco and Mei Zhen, developing this system that has been so fruitful for him, looking at a new whole other axis of complexity in the C. elegans nervous system, this notion of alternative splicing, where molecules can take on different forms, cell to cell, and mapping it and understanding it and making progress that way. So did that as a Bauer Fellow, is continuing to do that in his own lab at the University of Toronto, where because we failed to to retain him in this department 2 years ago when we offered him an assistant professorship. His Canada called him back. But it's my intention to periodically bring John back here as often as we can to maintain that connection and continue and begin new collaborations, because without overstatement, there's no one in his generation of C. elegans neurogenetics that I would rather work with as a colleague, and Canada's not that far away, so we hope to see you more often than this visit. So, without further ado, John.
Well, thanks, Arabi, for that really touching introduction, and the feeling is very mutual in terms of people in the C. elegans community that I would prefer to interact with, especially those that are always pushing the envelope in terms of systems neuroscience, behavioral imaging, which is something that we're becoming more and more interested in, and I hope that some of this work is going to eventually lead into that. That's at least the sort of 5-year plan, so I'm hoping maybe to come back in a few years and actually talk to you about systems neuroscience questions. But for today, it'll be about gene expression, and hopefully it's linked towards neurophysiology. Okay, so the title comes from Stanley Kubrick and 2001: A Space Odyssey. I'm a huge Stanley Kubrick fan, and so there'll be some slides interspersed throughout the course of this talk. So ever since I started venturing into molecular biology and picking up the right tools to sort of address the right questions, I've been interested in questions of gene regulation. In particular, what caught my eye was this sort of middle component of the central dogma, which is RNA. And we know now that RNA is quite a complex molecule. It's not just a go-between between DNA and protein in the information transfer in the cell. In addition to simply being transcribed, messenger RNA has to be capped, spliced in various formats. It can be cleaved and polyadenylated in different ways. There's RNA editing, modification, RNA can be exported and localized in unique and interesting ways. mRNA can be regulated at the level of translation, and then right up to the death of it at the level of decay, you can also have extensive regulation. Not only that, RNA doesn't always have to just encode protein. So there are a slew of non-coding RNAs, both short and long, that we've now come to appreciate. Some of the classical species were the ribosomal snRNAs, tRNAs, but then there was a huge explosion of data that started to suggest that there are these small and large non-coding RNAs that have implications in regulating both when and where genes can be expressed. And so we're always thinking about RNA regulation in all of these different layers. The other question that's fascinated me for a long time has been nervous system diversity. And so we've known for over a century from these beautiful stainings of Cajal that neurons come in all different shapes and sizes. And so really trying to understand this type of diversity in the modern climate, we need to really start understanding gene expression and how differentiation, development, and physiology is shaped at the level of these individual neuronal cell types. And there are a lot of people here that have been really at the forefront of some of the single-cell RNA sequencing methodologies and analyses pipelines that have been developed to start gaining an appreciation of diversity. And so we're trying to think about the interplay between gene expression states, post-transcriptional gene regulation in shaping both development, differentiation, and physiology of neurons. For the longest time, I've been interested in this particular layer of RNA processing. This is alternative mRNA splicing. It's a process where differential exon choice or splice site utilization can be utilized to create multiple outcomes at the level of mature mRNA species. And this can be regulated both in space and in time. And you can appreciate that one gene can make at least 2 products. But in the nervous system, we have some exaggerated examples of this. In the fruit fly Drosophila, for example, the dsCAM locus can generate upwards of 38,000 different variants. That's more than twice the number of genes in the fruit fly genome. And some really beautiful work from Larry Zapersky's lab has shown that you need at least a few thousand of these isoforms to properly wire a fly nervous system. In mammals, the equivalent exotic splice sort of locus are the neurexins, where some really nice work from both the Sudoff and Scheifele groups have shown that there could be upwards of 1,000 different neurexin variants, and these may play a role in specifying synaptic choice. So central to all of this regulation, and I'll speak about the sort of other half of what we're thinking of, are the RNA binding proteins. And so pretty much at all of these different layers, both in the nucleus and the cytoplasm, RNA is very rarely naked. It's often coated and decorated with these sequence-specific or sometimes structure-specific interacting RNA binding proteins. And they're pretty much involved in almost every transaction. And so the nervous system exhibits high levels of alternative splicing, likely due to the combinatorial action of these RNA binding proteins. Moreover, after the mRNAs leave the nucleus, there are ribonucleoproteins that have to be localized, and these transcripts can be subjected to both localized RNA locations and then locally translated, as I mentioned before. And then similarly, degradation can happen, and at all stages you invoke these transacting factors. So eukaryotic genomes encode hundreds of these factors. Each RNA binding protein can regulate hundreds of mRNAs. Some are more specific, some are more promiscuous. And every mRNA is generally regulated by, you can think of dozens of RNA binding proteins. Okay, so some of the key questions that we're trying to address are the mechanisms that enable neuron-specific splicing or even tissue-specific splicing. And this can be now extended to, in general, various layers of post-transcriptional gene regulation. The lab is starting to expand into other branches. How do repertoires of different splice variants actually shape neuronal function and neuronal development? And then how do these networks of regulated exons actually evolve? So a very interesting question is, how do alternative exons get incorporated into genes? How do they become tissue specific? And how does that sort of coordinate with biological pathways or understanding of how existing pathways are evolving? And again, we think about this from the perspective of the factors that recognize RNA, their repertoire of target transcripts, and ultimately the consequences on phenotype. And this is sort of analogous to the way people think about transcription factors, right? You have sets of transcription factors that are likely regulating a coordinated set of targets, and then those targets have some influence on aspects of biology. For the last sort of 5 to 7 years, I've been using this beautiful nematode, C. elegans. I can never get enough of it. I keep saying that I want to get back into mammalian work, but I always find new things to do with this model, and that's because there's a lot of really useful tools available. So it's genetically tractable. It exists as a hermaphrodite. Its life cycle is every 3 days. It has a simple, but I should say compact nervous system rather than simple. 300 neurons. Most of the major classes that are found in mammals can be found at least at the level of neurotransmitter type or neuromodulatory type. We know the synaptic connectivity for some time, although it's being refined by efforts here by Aravi's group in collaboration with Jeff Lichtman and Mei Zhen in Toronto. So there's a really nice neuroanatomy that's constantly evolving and creates a really great landscape for us to look at aspects of neurobiology. And the cell lineage itself has also been completely mapped, and we have a lot of really great markers that can label. We can drive transgenes, and even in single pairs of neurons. And so this is a really great system if you want to start asking questions at the interface of neuronal differentiation and then going back into a model system to do in vivo work. And that's really what attracted me to this model, because we have the ability to rapidly jump in and out of multiple scales and asking questions all the way down to molecular work, right up to organismal behavior and physiology. Okay, so today what I'd like to chat with you about is some of the ongoing work where we have been making a number of genome-wide observations of tissue and neuron subtype-specific alternative splicing in C. elegans. And then pending some time, hopefully I'll give you a brief overview of a sort of ambitious and project that's still in its infancy, where we are trying to characterize the RNA binding protein expression landscape at single neuron resolution in this nematode. Okay, so this project started actually while I was a fellow here, so really talented postdoc, Hiko Gracida in the lab, who's still here with Yun Zhang, helped develop and really cement this translating ribosome affinity purification technique called TRAP. And so the way this approach works is you can generate a transgene where you have a GFP-tagged ribosome subunit. In worms, it's called RPL1. In mammals, it's the L10A subunit. What you can then do is drive expression of this transgene under the control of specific promoters. Then you can just make a mashup of whole worms, many animals, in the presence of cyclohexamide. And what this does is it arrests the ribosome on its actively engaged mRNAs and creates a very strong sort of high affinity interaction that you can then use sort of semi-stringent purification schemes to enrich for your sample of interest. And what we do is we use antibodies that had been developed by Miriam Heyman when she was a postdoc in Nat Heinz's group at the Rockefeller to purify these transcripts. And so the idea is you can get a transcriptome enrichment of the actively translated mRNAs from a given cell or even neuron type. We then perform quality control experiments to look for enrichment and function, sort of made sure that our IPs work properly using just a simple RT-PCR assay with some marker genes. And then, of course, we move on to making sequencing libraries and computational analyses to look at both gene expression and splicing, which is the bulk of what we're going to speak about today and the rest of this first half of the talk. Okay, so these are the strains that Hiko had played an instrumental role in developing in the lab. So this is our paneuronal strain. This is the head of the animal, ventral nerve cord, and tail. This is the body wall musculature of the animal. These are the intestinal cells here. And then we can also, as I mentioned, label subtypes of neurons. So we have the 8 dopaminergic neurons and the 6 serotonergic neurons labeled in these animals. So we don't have access to a HAL 9000, but fortunately there have been a lot of different algorithms that have come on board for people like us who are not exactly computational tool development people. So we've been using STAR for our initial mapping of the transcripts back to model transcriptomes in the genome. And then for analyzing local splicing variation and splicing connectivity, we were using a pair of packages called Magique and Voila from Yosef Barash's lab, who's a colleague. Okay, and then when we actually started looking at the data, we sort of got a little carried away, I think, but it was great to actually sequence quite deeply. So we have paired replicates of each of these broad tissue types and neuronal subtypes. We've generated over 1 billion uniquely mapped reads, and we have whole animal lysate transcriptome. So this is what goes into the IP in matching sort of depth, so that we have almost another 1 billion reads of just whole animal prepared transcriptome data sets. Okay, so the first thing that we wanted to ask with this data was whether or not we could actually start to see novel splice junctions identified that we couldn't otherwise see by using whole animal data. And so we took our whole animal data set, tried to identify splice junctions, and then started looking across our tissue IPs and asked whether or not we could actually detect novel splice junctions that hadn't been annotated or couldn't be detected in our data. And we gratifyingly and maybe not surprisingly start to see splice junction reads that have pretty decent coverage, so they're not really one-off types of reads. We can extend our read support up to as much as 25 reads and still retain a number of splice junctions that we wouldn't otherwise see in our whole animal data. So this alone, from a point of view of annotation, is already creating a wealth of information that we wouldn't otherwise have had if we didn't have these tissue biased or cell type biased transcriptome data sets. We wanted to extend this analysis a little bit further, so there was a recent metadata analysis published a couple of years ago from Denis Dupuy's group, where they analyzed over 1,000 different worm whole animal transcriptome data sets and came up with a list of junctions that I think totaled a number of over close to 700,000, so both constitutive and alternative junctions. And so we just compared our whole animal data with that, and it looks like overall, it was gratifying to see that the splicing patterns and the relative junction usage, so we use this percent spliced in metric, where 1 means the exon is always present in a given transcript, and 0 means that the junction is almost never detected relative to its neighboring junctions. So overall, our whole animal data set agrees with, on average, 1,000 whole animal transcriptome data sets. But when we actually start to analyze our tissue IPs, this is where it starts to also get interesting. So even if you have 1,000 pooled whole animal transcriptome data sets, you still do not get any additional sampling power that you would otherwise get if you even had sort of 100 million reads with individual tissue profiling experiments. So we start to see different branches. So these are sort of intestinal and neuronal splice junctions that just are not detected in other cell types or in whole animal data at extremely, extremely high sampling and repetitive sampling. Same thing with the neuronal subtypes. We also see branches of splice junctions that we can see that we wouldn't even pick up in our whole or broad neuronal sampling. So we think, again, this is sort of maybe preaching to the choir here, but as we start refining our transcriptome data, we're starting to appreciate new transcript architecture and junctions that are being utilized that wouldn't otherwise be found in some of these other data complex or heterogeneous data sets. Okay, if we start to actually compare the IP data against one another, we can start to also detect differential junction usage between our broad tissue types and also between the broad nervous system and the two different subtypes. And one of the things that sort of struck us with this analysis was that the nervous system, much like what's been advertised in other invertebrates especially, seems to stand out as a tissue or organ system that has prevalent differences in splicing patterns. So if we look at these clusters and heat maps, we can see that there are always often patterns that emerge in the nervous system that are distinct from, say, the other two tissues. If you start going across, there's another pattern here that very quickly emerges. The one thing that was more surprising was actually this sort of broad nervous system comparison to the neuromodulatory neurons. It looks like, again, the broad nervous system seems to be largely having a distinct splicing profile from these neuromodulatory cells, and we're not quite sure what to make of this yet. One theory that we have is that in sampling the full nervous system, the most predominant neurotransmitter type of cell are cholinergic cells in the animal, so it's possible that what we're seeing here is a distinct splicing pattern that's also emerging between cholinergic subtypes and these neuromodulatory cells. We have to do a bit more digging into the data to sort of flesh out some of these results, but this is one interesting and maybe surprising observation that we've seen. So if we look at how it breaks down, similar to what I just described, if you start to look at comparisons, again, the nervous system seems to be dominating in terms of the number of changes that we find. So the Venn diagram here, if you look in the blue and yellow areas, are the nervous system versus everything else. And so perhaps not surprising, it's the most diverse set of cells in the sampling that we've undertaken. Maybe this is what's contributing to its distinct profile. So now that we've identified a number of robustly different alternative splicing events between these tissues and subtypes, we now want to actually start asking some questions. Can we learn anything about the features of these types of exons? And this is what's going to be coming up in the sort of remaining slides for this section. So if you perform a simple Go enrichment analysis, you can start to see that a lot of these differentially regulated exons more often land in genes that are involved in protein signaling cascades, ion channel physiology and function, and perhaps rearrangement or activation of the cytoskeleton and cytoskeletal dynamics. And so we're really interested in starting to look at some of these gene sets and maybe figure out where these exons are actually landing, what functional output they might or consequence they might have on the nervous system, and whether or not even within the nervous system there's diversification at the level of transcript usage. Okay, the other thing that we wanted to do is a sanity check, because the trap technique hadn't really been established in the worm, was to sort of use an independent method to try to validate some of our observations. So we had these two-color splicing reporters that many of you had probably heard me speak about. They've been a workhorse in the lab, but what they employ is, we can take a splicing event of interest where the middle exon is alternatively spliced and clone it in as a mini gene. So we just take the 3 exon 2 intron portion of the gene and put it upstream of 2 fluorescent protein open reading frames. One encodes green fluorescent protein, the other is M-cherry. And we can then introduce a single nucleotide into the alternative exon and allow it to shift reading frames, such that when the alternative exon is included, you get production of a linker peptide that is not fluorescent in the GFP open reading frame, but translates into the frame with the M-cherry protein. So you get production of a red fluorescent protein. If the exon is skipped, you get production of GFP. And so we couple this with a nuclear localization signal, and you can really start to see dots within the nervous system or any other tissue of the animal and really start to visualize alternative splicing. It's a great proxy for what's happening at the level of the RNA splicing choice, and we can get down to single cell resolution with this. So this is one example measurement. So there's a gene called HLB1, which is an ortholog of Liprin beta, which has an important function in organization of neuromuscular junction and function. It's required for wild-type locomotion in worms. And so our measurements for TRAP suggested that the muscle cells were 100% included with this, in terms of their expression of this alternative exon in transcript populations, whereas in the nervous system we have complete skipping. And so when we make reporters and start to look at these in the animal, we can see, again, the green channel is representing the exon skipped pattern, and the red is representing the exon included. And we have co-injected 2 reporters where all we've done is changed the promoter that's driving the expression of the same reporter. And if we actually look at the sort of location and shape of the cells, and based on the anatomy, we know that these green dots here are central nervous system neurons. These are ventral nerve cord neuronal nuclei. And so as measured by TRAP, there's extreme exon skipping in the nervous system. If we look at these cells here, they're the muscle cells. These are the body wall muscle cells. And so in the muscle, you have predominant use of this red isoform. And then the overlay very nicely shows how those 2 patterns are emerging. So we're really starting to be satisfied with the transcriptome analysis. We think that by using this completely independent method and starting to see validation, that we can trust sort of what we're looking at for the most part. Okay, so for the remainder of sort of trying to look at the features of these exons, we had to create sort of different groupings of exon type. And so we created these different categories and grouped our data according to these classes. So we found exon triples that had reliable detection in our transcriptome data that represent what we call constitutive splicing events. So these are exons that are always included 100% of the time in all transcripts in all cell and tissue types according to our transcriptome data. We also have alternative exons, which are we call non-tissue differential. So these are exons which have sort of roughly the same proportions of usage in the population from cell type to cell type across at least what we've measured in the data. And then finally, we have a group of tissue differential events where we actually have now measured using TRAP variation or significant variation that's reproducible across some of the cell and tissue types. And so we can start to ask questions about various features of these groupings of exons. And so the first thing that we asked was whether or not these different groups of exons are more likely to preserve reading frame. So are they a multiple of 3 or are they not? And so when we look at constitutive exons, they are less likely to preserve reading frame. This makes sense because it doesn't actually, if you're always splicing the same way and you keep your exon size the same across evolution, you'll never disrupt a protein domain because you're always splicing it the same way, and so there's no need for achieving frame preservation. However, if we start looking at the classes of alternative exons, we see now that there's an interesting bias towards having to preserve frame. So it really suggests that these exons are likely inserting or removing bits of protein sequence. And we see maybe a slight statistical enrichment of these tissue differential exons. So there's even more of a drive when you have regulation across tissues to really preserve or be a multiple of 3. If we start to look at overlap with protein domains, this is something that's a hallmark of exon evolution also in vertebrate studies. So we're seeing a similar type of trend in C. elegans and some of the related nematodes where constitutive exons tend to more often overlap with protein domains. Both the alternative exon classes tend to actually avoid protein domains or have less overlap with protein domains. And at first we were I think the field as a whole was disappointed in this result because we initially thought, well, alternative exons are going to be modulating protein-protein interaction domains or enzyme physiology or function. But there's been a new sort of movement in the field towards thinking that some of these exons are actually overlapping with what are called low complexity, intrinsically disordered regions, which also have the ability to influence protein-protein interactions. They've become more in vogue over recent years because of these phase transitions that have been discussed, or I like to call it blobology. I think we still don't quite know exactly what, in some cases, what these things are responsible for. But we're trying to do that analysis now, and my guess is that these exons are going to more often overlap in some of these types of intrinsically disordered regions, much like their vertebrate counterparts. Okay, we can also start to look for motif enrichment. So in terms of getting back to understanding mechanistic function, so we've made a number of different permutable comparisons. This is just one of those comparisons that I'm showing just to highlight this. So we're using a software package called Homer that will take linear motifs and look for overrepresentation when you compare one group against another group. And so sure enough, when we do this type of analysis and focus on the region that's historically been associated with splicing regulation, the exon itself and its neighboring intron sequences, we start to see enriched motifs that are overrepresented in classes of tissue-enriched splicing events. And some of these are very well studied and well-established motifs that are recognized by known regulators of splicing and play other roles in RNA biology. So HNRNPL, the CELF family of factors. FOX1 is a very one of the first RNA binding proteins where the motif was actually known and sorted out using bioinformatic methods. So it's been very gratifying to see that we're starting to pull out motifs of factors that we've been working on and other people have been working on in the field. There are also a lot of orphan motifs that share equivalent enrichment patterns that we're really excited to try and figure out what they might be what might be the cognate RNA binding protein that's recognizing these factors. And so a branch of the lab is now focused on building reporters, doing mutagenesis on these motifs, and trying to sort out whether or not they indeed are causally important for regulation. So this type of analysis can really start to lead to interesting questions. Okay, the last thing that I'll show is just, again, some simple questions that we're asking, but we're seeing some very interesting results. So we asked whether or not the length of the introns surrounding some of these exons are actually, different, or they're distributed in a different manner. And it turns out that, the worm genome, unlike, vertebrate genomes, is much more compact. And so on average, intron length is actually quite small. So the median intron length of our constitutive exon data sets is about 50 bases. There's been about a fourfold expansion in size at both the, whether or not you're differentially spliced across tissues or not, but as long as you're an alternative exon, your intron size tends to be a lot larger. And so we're not quite sure yet what to make of this result, but it's sort of interesting to think of. Some people have thought that by lengthening the size of an intron, you might weaken the ability to splice efficiently, and that allows for regulation to sort of evolve and emerge. The other possibility is that having a larger intron might allow you to accommodate more regulatory sequences, although we need to sort of do a bit more work on figuring out which of those two possibilities might be at play. Perhaps they're not mutually exclusive. Okay, the other cool thing was that these alternative exons tend to be shorter than constitutive exons. So if we look at the distribution of both the non-tissue differential and the tissue differential events, they're really skewed towards having shorter exon sizes. And this was interesting because when we actually look at the distribution, I was sort of looking at everything a little bit more closely over the last few days in preparing this seminar. About a quarter of the exons are actually less than 30 nucleotides in size. And so this is really interesting because in the field, there's been this, emergence of a definition of exons called microexons. I'm not sure, they've always kind of been there, but somehow now by giving them a name, people have started paying a bit more attention. I think the idea is that if these exons are very tiny, it's actually really hard for the spliceosome to assemble across the splice sites of the exon. And so the thought is that these have to fundamentally be regulated in a very different and distinct way from standard exons. And interestingly enough, these microexons seem to have a prominent role in the nervous system of vertebrates. And a lot of more recently evolved, or I guess recent to us, RNA binding proteins are thought to be specially defined in regulating these microexons. And so it was actually kind of surprising to see that there are quite a few differentially regulated exons in the worm that are actually this small. Here's an extreme example of one. This is a 9-nucleotide microexon that, so I'm just showing a conservation track with, it's a multi-species alignment of surrounding Cinerabdidus genus nematodes. And although these worms look the same, they actually are about as diverged as human and fish. So it's actually a quite diverged set of species in terms of substitution rate. And this exon is extremely conserved, and the surrounding splice sites and neighboring cis regulatory elements in the intronic regions are also very highly conserved. And so usually when you see things that are this highly conserved, it means that they probably are functional, although trying to assess the function of inserting 3 amino acids into a protein starts to become very difficult. But we're starting to take on this task of making CRISPR knock-ins to address this. This is one example where we have made a knock-in, and I'll show a bit of preliminary data with this, but this is an 18-nucleotide microexon in an ortholog of the APM2 gene. So this is an adaptin protein that's involved in clathrin-mediated endocytosis. So we think maybe there might be a connection between some of the splice variants and synaptic vesicle dynamics, recycling, and synaptic vesicle biogenesis. And so this exon, again, is fairly highly conserved. The surrounding splice sites are highly conserved. We did what any self-respecting geneticist in the modern age would do. We started making CRISPR knock-ins. So we've engineered ways now to forcibly express one isoform or another in mutant animals. So we can edit the loci to make sure that only one splicing outcome occurs. And so this is the wild-type locus. It's hard to see both isoforms. It looks like the microexon minus isoform predominates in whole animal samples. But then you can see that very clearly we can now force expression of only the microexon plus form or the microexon minus form. And now we can ask some simple questions on whether or not presence or absence of this microexon in the genome has any influence on aspects of physiology. So we're getting better and better at doing neurobiology experiments and behavior experiments, but the first place to start is this very simple assay which we developed a little while back, where we can co-incubate worms on the same plate and allow them to sort of compete for the same resources. And so we put an equal number of fluorescently labeled animals with non-fluorescently labeled mutant animals, and we ask whether or not one genotype can outcompete the other, whether there are any fitness defects. So at the end of the day, you can measure what's called a relative fitness phenotype, where you can just count the percentage of mutant animals and divide that by the percent expected if there was sort of no fitness defect, which would be 50%. And so when we do this experiment, we can see that our control GFP strain, when we compete that with the wild type strain, relative fitness is 1. That's to be expected. If we take these microexon containing only animals and compete them against the wild type mark strain, again, fitness doesn't really change. We take these worms that now have lost this microexon, and we see that there's about a 15% fitness defect in these assays. So this doesn't seem like much, but remember, we're just getting rid of 6 amino acids from a protein, and so 15% in the wild would probably kill you by the end of a couple of generations. So we're very intrigued with these initial results. We would love to start deleting and targeting more of these highly conserved small exons and try to figure out how they may even work together to create additive or complex genetic interactions. Okay, so just to summarize, what I've told you in this first part of the seminar, we've shown, I think, pretty well that cell type specific transcriptome profiling can identify interesting novel splice variants across tissues and within neuronal subtypes. We can start comparing these types of exons and learn some features about them. So these alternative exons tend to have increased frame preservation, longer introns, but shorter exons, including the emergence of these microexons, which were thought to play more of an important role in vertebrate biology. They tend not to overlap protein domains, so we're thinking about this in the context of these unstructured, disordered, low complexity regions. They certainly harbor motifs for known splicing regulators, but there's also these novel motifs that we would love to pursue. And it looks like they are enriched in certain classes of biological processes. So we're also interested in targeting some of these processes and maybe looking at these exons as a group or a module. And so that is really a long-term goal stemming from this, this infrastructure that we've developed in terms of starting to delete not just one exon at a time, but maybe combinations of exons in defined pathways and see how additively or synergistically they may have an effect or impact on neurophysiology. And then, of course, some of the work now in starting to look at conservation features is the emergence. So like the birth and death of these exons has become very interesting to me. So we're seeing all sorts of spectra of conservation patterns. There are some that are very, very ancient, these exons. Others are more recently emerged, but are still across several sister species when we look at the sequence alignments, and so we're really, really fascinated in how these dynamics are playing a role in shaping these biological outputs. Okay, this is the part where it gets a little bit sort of weird, and so this part of the movie was oftentimes preferred to be watched alone and in the dark, and so it'll be, I guess, visually appealing. There will be very little dialogue, much like in the movie. So it's just, the idea for this portion was just to sort of show you some images that we're starting to observe, and maybe you can all help me try to figure out what is going on. We're hoping to figure this out over a long period of time, but there's nothing like doing microscopy in terms of discovering new biology. And so that's what the goal of this part of the project that we've initiated in the lab. So a talented postdoc, John Laver, has really taken this on and has really flushed out a lot of interesting results already, but we still have a long way to go. So in an ideal world, I guess the motivation for this project is that, I mentioned that oftentimes a transcript can be coded with dozens of RNA binding proteins. And, if we really want to try to understand how these factors are contributing to, mechanisms of regulation at the post-transcriptional level, we need to try to figure out exactly where they are, what subcellular compartment they're in, are they in different types of foci, and once we maybe know that, we can gain a slightly better appreciation. I'm not saying we're going to still learn everything. There's always going to be stuff to figure out, but it might help facilitate modeling this regulation. So we would like to start working with computational folks to start building better models of how this type of regulation can transpire when you have collections of RNA binding proteins recognizing different sequences in different space and time. And also, I think, especially in light of single-cell RNA sequencing, I think a lot of people have sort of mentioned to me, why not just do it that way? And we're certainly being informed by all this data that's being generated. But I think at the end of the day, you miss sort of a layer of information, which is, where are these proteins actually localizing to? So it's one thing to know whether they're on or off, or present at 50% at the transcript level, but where they actually physically located is conveying a lot of information in its own right. So it's lower throughput, but hopefully I'll be able to show you that it's worth doing. And so the approach at the beginning has been to just obtain a library of GFP tagged RNA binding protein transgenes and just ask simple questions of, are these transgenes expressed in some neurons and not others? Are they expressed everywhere? Are they only expressed in a very select set of neurons? And so fortunately, Misho Sarov at the Max Planck Institute in Dresden had developed this FOSMID library where they've tagged essentially 80% of the genes in the C. elegans genomes at the C terminus of the encoded protein product. And these FOSMIDs contain the gene of interest in the context of 40 kilobases of DNA. And so from a worm perspective, that means most of the endogenous regulatory signal, not only of the gene, but also its neighbors. And so we really think that this is probably the best way to assess, at least transgenically, what the endogenous pattern is, while still having it in a modular manner and in an existing format such that we don't have to do maybe CRISPR editing for many factors, at least at the onset of this project. The other plasmid that we co-inject into the animals is a reporter that just labels all neurons. So it's a pan-neuronal marker that expresses a far red protein called Neptune, and we're just simply looking at overlap in expression. So the idea is to assess whether or not, as I mentioned, these RNA binding proteins are expressed in particular neurons, whether they have different cytoplasmic or nuclear localization patterns. Can you see it in neurites, for example? And so this is just a walkthrough of some of the things that we've started to see. So this is an RNA binding protein called that's part of the TIA family of regulators. So they have important roles in both splicing and translation. And so again, if you look at the magenta signal, this is representing whatever neurons we could pick up in these sort of mosaic animals. But the idea is that every cell should roughly have both plasmids in it. So you can really start to look at questions of whether or not there are red cells that are no longer green, and try to assess that. And I think you'll appreciate in the overlay here that we can already start to see that there are clearly some neurons that do not express this RNA binding protein. So this is one of our sort of nicer examples of this. And it looks like this factor is predominantly nuclear in its localization pattern. This is one of our favorite genes right now. This is our most selective expression pattern. So this is a quaking ortholog that in mammals was initially characterized as having a defect in axon myelination. In the worm, there are actually several paralogs of this factor, and this is one of them, and it has a very select expression pattern. So we see it mostly cytoplasmically, and it's expressed in 3 pairs of sensory neurons. And I'll get into which ones they are in the coming slides. This is another example of an expression pattern taken at much higher resolution. And another feature that we're starting to appreciate with some of these RNA binding proteins is that different cells are expressing the factor according to different localization patterns. So in this image, this micrograph alone, we can actually start to see 3 distinct localization patterns. So there are non-neuronal cells which are not being labeled in red that are exclusively cytoplasmic in terms of their localization pattern. There are other neuronal nuclei that seem to have a more sort of dispersed nuclear localization pattern. And then we also see these sort of discrete foci or puncta that are are forming in some of the other cells in the nucleus. So what's really becoming apparent in a lot of these localization patterns is that even the same RNA binding protein, depending on which cell it's in, might actually manifest its output in a slightly different way. In some cases, it might be in the cytoplasm and exerting function there, and in other cases, it might even be in different domains of the nucleus. And so what we're really interested to understand right now is whether or not these factors, or these localization patterns, are always the same across every animal in the same neuron type, because then it would suggest that there's perhaps active regulation on a neuron subtype level that's causing these patterns to appear. This is an interesting one. So this is the ortholog of dicer. So it's one of the key dicing enzymes involved in some of the small RNA biogenesis pathways. We're not sure what to make of this, but this is another case where we're starting to see this factor clearly expressed in the nuclei of some neurons in the nervous system. We also have this very bright cell over here where it's clearly a cytoplasmic signal. This is another cell here that's overlapping with the neuronal nucleus that it's clearly cytoplasmic, and dicer is getting out into the dendrites all the way to the tip of the nose. So some really crazy and weird expression patterns, and so we're very fascinated about some of these results and what to make of them in the context of maybe some of the recent results where people are implicating small RNA biogenesis pathways in neuron to germline communication and transgenerational phenotypes. So maybe this is a way of conveying information perhaps across generations. And maybe by modulating some of these small RNA biogenesis pathway enzymes, you can actually affect how the environment is speaking to the organism, right? And how that information can be transmitted even to the progeny in the next generation, or even within a generation across cell types. Okay, so far we've looked at I won't show you all of the aesthetically pleasing images, but we have about 40 RBPs that we've examined in this manner, and we have another 60 that are in the pipeline, and that's going to represent a significant portion of the RNA binding protein space, at least in terms of what we determine to be somewhat enriched or at least quite highly expressed in the nervous system. And we're starting to see some trends emerge. So it seems like more than 2/3 of them look like, to a first approximation, they're in almost all neurons that we see. So there isn't maybe as much subtype level expression patterns. The other third are in what we call neuron subset specific expression patterns. We only have that one example where we only saw expression in 3 pairs of neurons. Most of the patterns look more like the TIA example that I showed. So you have quite a few neurons that are expressing, but there are some subsets that are absent. And so we think this might be a distinct type of organization from, say, some of the more sparsely encoded transcription factor expression patterns that people have started to see in different model systems, including in the worm. Some of the work from Oliver Hobart is suggesting maybe there are these sparsely encoded transcription factors that are shaping neural differentiation. With RNA binding proteins, it's starting to look as though there's more, maybe the points of overlap are becoming more important rather than sort of having one RNA binding protein that specializes maybe 2 or 3 neuron types.
So have you selected RBPs that were already known to be neuronally expressed?
Yeah, I didn't speak about that. So we've tried to enrich the data, at least to prioritize the list on our TRAP-seq data. And now we're also incorporating single-cell RNA-seq from Bob Watterson's group and other people who are starting to generate this across different developmental stages to at least assess whether or not some of these RBPs are detected in the nervous system. Yeah.
There must be RBPs that are like muscle-specific.
That's right. Yeah.
Not
Not
looking at those.
right now, no. Yeah. Based on the slide in the introduction, it's always been the nervous system for me. But yeah, I agree. It's a great unexplored area that we should maybe this would build upon future projects where we should just look at every cell in the animal if we have enough time, resources, and people to to do this type of stuff. And then the other interesting point is that roughly in equal thirds we have factors that are nuclear, cytoplasmic, or shuttling, or present in both compartments. Sometimes in the same cell, other times in distinct cells. And so this is really starting to create this complex picture of localization. And so we're really, really fascinated in trying to flesh some of this out. So a big question now is whether or not these patterns, as I mentioned before, are always in the same neuron, different neurons. Is it random? Is it stochastic? And so to do that, we really want to be able to put our money where our mouth is and really be able to create this type of atlas where we can really say this RNA binding protein is expressed in this neuron. And so fortunately, over the last few years, there have been a number of groups that have been working to develop what we call, I guess, a static brainbow in the worm. So sort of, taking from inspiration probably the work from Josh and Jeff here, but wouldn't it be great if we had a line that could actually light up every neuron slightly differently, but only could it be every every animal has the same neuron, the same color, right? And so Ev Yamini, in what's been a sort of tour de force over the many years from Oliver Hoberstab, has generated this strain called the NeuroPal, a neuronal polychromatic atlas of landmarks, where they use 41 different driver transgenes expressing different promoter combinations with 4 different fluorescent proteins. So TAG-RFP is a pan-neuronal marker, and then there are these 3 other proteins which are called landmark colors. And they are deliberately chosen to be orthogonal in spectral properties to GFP, such that you can image the strain and then overlay on top of that a GFP expression pattern. And so Oliver's group, and in collaboration with Aravi's group, particularly Albert Lin here, has done some really beautiful work looking at sort of brain-wide imaging patterns using this strain and trying to, again, associate different neuronal activity patterns with the identity of the neurons themselves. We're using it just simply as a tool for identifying neurons. So this is what this strain looks like. So these are neurons in the head, the ventral nerve cord, and tail. You can see immediately, even at this high, sort of low magnification, that it's really A, beautiful to look at, and B, very informative, because it really does appear that you can sort of identify the neurons. And so Ev has done a really nice job. He has a nice sort of instruction manual for the novice neuroanatomist to try and really associate colors with different neuron types. And so we're in our infancy, sort of as of, I guess, the last few months of sort of navigating both acquiring images with this strain, and also now annotating the actual neurons themselves. So it's been a really fun thing to start doing. So of course we went after the easiest expression pattern that we had. This was this factor that was only in 3 pairs of neurons. We said if we can't do this, then we might as well retire and not embark on this project. And so it turns out that it was actually fairly straightforward to sort of make the sort of double transgenic animal and then overlay the neuron patterns. And we found out very intriguingly that these 3 sensory neurons turn out to be ASC, AWC, and AFD, which are very interesting neurons because they are involved in a number of different environmental modalities and behaviors. So you've probably heard a lot about thermosensation from talks from folks in Aravi's lab. AFD, among other things, is also implicated in carbon dioxide detection. ASC is more a class of neurons that detect soluble attractants like salt. AWC has been a very well-studied neuron involved in many different biological pathways and behaviors and sensing of volatile odorants, pheromone detection, thermosensation as well to some extent. And then I didn't mention this, but actually there's some data that we have from animal to animal suggesting that occasionally we see this other neuron pair, this ASK, which is another interesting neuron that's thought to be involved in pheromone detection, dower entry, and also sexual attraction in males. And so when we went back to the single-cell RNA-seq data that had been collected, it turns out that ASK turns on the expression of this factor a little bit earlier in development, and then it kind of shuts down. And up until now, we've been imaging late larvae, where I think the expression is just getting turned off. And so it's interesting because we may also have a developmentally regulated expression pattern here. And so that's another question altogether. So far, we've only been looking at mostly close to adult animals, but how these patterns are going to be changing across developmental time is another sort of unexplored area. So that's really, I guess, all I wanted to speak about. I don't have a summary because I kind of, again, it's still very observational, but hopefully you can take an impression of what we're already starting to see with this data. I think there's going to be a wealth of information to be gained, and then of course, starting to figure out what some of these things are doing So we have a knockout of this animal now, and of course it looks totally normal, at least superficially, but we're now starting to get people like Aravi's group to start looking at maybe some effects on behavior, neuronal activity, and we're just trying to figure out which modality to start testing. Of course, I wish it was only one type of modality, but it looks like there are a number to choose from, so we'll have to see what happens. Okay, so I just wanted to thank people that we've been collaborating with over the year. I mentioned Hiko, who has been continuing on some of this profiling work in Yoon's lab. An undergrad at Harvard, actually, Taylor Shirtliff-Hines, who is in the LS50 course, but is a Torontonian, came to work with me one summer and actually created those dumpy 23 microexon CRISPR knock-in strains. And so she was instrumental in generating that data. So I hope she had a great summer. Of course, Oliver and Ev for providing us with the NeuroPal strain, Michel Sara for help with obtaining these phosmid transgenes. Yoon and Aravi's lab, of course, for being continuous collaborators and colleagues in some of these exploits and teaching us about neurobiology. And we are still, I guess, gene expression people trying to moonlight as neurobiologists, but having people like that to interact with have been great. And then, of course, people in my lab. So Bina is the person that did a lot of this transcriptome analysis, all the informatics that I showed you. Pallavi has been working on the evolution of splicing and these conservation patterns that we're starting to see, trying to do multi-species alignments. And John Laver is a really talented postdoc that has been, has taken up this RBP localization atlas project. So thanks so much for your attention. And if you have any questions, I'm happy to take them. Yeah. Hi. Yeah.
From the first part of your talk with TRAPSeq, if you found any evidence of backsplice junctions that you would find in circular RNAs, have you done any analysis?
That's a great question. We haven't looked at all at backsplicing. We really should try to do that. I think over time we've been adding new things to look at in the analysis, and that's something that we really should. And of course, there are circular RNAs in the worm that have been characterized in other experimental sample sets, and so it would be really interesting to start looking at that type of stuff. Yeah. And that would be the other thing. Yeah, there is this thought of that some of them might actually be substrates for ribosomes, and you can get what's called maybe circular translation, where the ribosome will actually just continue translating off of them. Yeah, that would be really interesting. Yeah. Hi.
Have you found any examples of left-right asymmetric or male-female dimorphic RBP expression patterns in neurons?
That's a great question. We have not looked at anything like that yet. I think that's something that we were just speaking about with even this very specifically expressed factor, to see if there are any male hermaphrodite dimorphic expression patterns. Left-right asymmetry, it looks like, as far as we can tell, some of these neurons are, like ASC is an asymmetric neuron, for example, and AWC also is but it looks like to our first approximation, it's present in both neurons. So at least in those neurons, it's symmetric. But yeah, we're hoping that with the NeuroPal strain now, we can actually start looking at things like left-right asymmetry across the animal. It's a great question. Yeah. Yoon and then Kristen. Yeah.
So when you look at all those RNA binding proteins, in addition to look at where they are expressed, so just based on their protein sequences, can you categorize them, or can you say that they may have several different types of function?
Yeah, so we haven't, we've characterized them by their RNA binding domain sort of family composition. So there are, representatives from all of the canonical RNA binding domain families. We haven't done, I think, what you're suggesting, which is to also see if there are other domains in some of them and see if we can group them. I mean, that would be very interesting to start doing once we have maybe more of the atlas kind of made, and maybe we can correlate whether certain families are overrepresented in being present in certain subcellular compartments or in certain cell types, or yeah, I think that's a really great direction we should go in. Yeah. Kristen. Yeah.
I was wondering if you, in
Yeah, we haven't, those are both really great things that we should look at, I think. We've only just started, I think when we generated that distribution pattern, it was always like, we had this data, we looked at it, and then now upon actually starting to do some quantitative analysis, this is sort of like 2-3 weeks old where we started seeing these like actually paid attention very closely at the fact that there are these exons that are so tiny. Judging at the anecdotal, looking at some of those, I've been spending a lot of time looking at these sort of multi-species alignments and kind of walking across, the neighboring exon-intron architecture. It looks like they are I'd say more highly conserved than your typical, even constitutive exon architecture sometimes. Like when they are there and they are part of these, tissue restricted or tissue biased groups, they seem to have these very old sort of patterns in terms of like in almost all 7 out of 8 or all 8 of the Cinerabdidus species that are in the current alignment, they're there. I mean, it's just crazy. Like the splice sites are well matched, and so to me that indicates that there's some function. Whether or not there's interesting things that happen in the neighboring exons as well that favor this type of emergence. So maybe it's like long introns or something. That would be, yeah. The correlation would say like all of these small exons are flanked by on average larger introns, but we have to start doing some of that. Yeah, yeah. Those are great questions. Yeah. Hi.
Do you see any alternative splicing in RNA binding proteins, and might that have any correlation with the differential localization you see?
Yeah, absolutely. I think that's sort of putting another wrench in the whole understanding of this. So yeah, with a lot of these transgenes, they're C-terminally tagged, and where they decided to put that tag sometimes may only label one isoform. The short answer is yes, there are a number of RNA binding proteins that do also have splice variants. Some of those splice variants are there for regulatory purposes. So the common theme with RNA binding protein splicing is that you have autoregulation and cross-regulation to help control the levels, such that you make too much of the RNA binding protein, you splice one way, and you start reducing down your levels, and then all of a sudden you go down below a certain threshold, and now you start to splice the other way because the RNA binding protein binds its own transcript. And so the one example that I showed, DIN1, actually John, my postdoc, had mentioned that there are in fact 2 isoforms, and so DIN1 was actually characterized. Its name stands for DAF12 interacting protein. So it's been originally annotated as a transcriptional cofactor, a repressor for a key transcription factor in the Dauer pathway. And the one variant has the DNA binding domain in it. There's a shorter variant that actually lacks that DNA binding domain, but maintains the RNA binding domain that's in the protein. And so there's some interesting. So one question we have is whether or not, so that transgene is labeling probably both of those isoforms. So we really want to know if only one of the 2 isoforms is responsible for maybe the nuclear foci versus cytoplasm. And so there's some very interesting questions. Yeah, it's getting very complicated very fast. Whenever anybody tells you C. elegans is a simple organism, it's more than complicated enough for me. Yeah.
Well, with that, thanks.
Okay. Thank you.